much, Janusz, and thank you to all organizers of this meeting. It's a real pleasure to be back to India, and it's not my first time to India, but it's my first time to Varanasi, and I'm really thankful for this opportunity and also the privilege to address you at the beginning of the conference. In my talk, I will focus on several concepts and words widely will be likely used in this conference. Terminology about genetic ancestry and relatedness, as we know, sometimes these words can have potentially be used in a negative context, and we really need to be clear where we draw the line between ancestry and ethnicity from one hand, or ancestry and race on the other. But before I start, I would like to also say a few words about the Professor Svante Pääbo, whose work we are celebrating here. So 2022 has been really a great year for all of us because he, even though he won the Nobel Prize in physiology and medicine, but we really know that he's representing evolutionary biology as well as anthropology, and therefore it is a bigger occasion to all of us. And also, as it was already mentioned, his contribution to the field of archaic genetics has been extremely important. But also when thinking back in time in 1990s, when visiting his lab in Munich, at that time ancient DNA was not an easy task to be undertaken. The way that now we see ancient DNA labs flourishing in every corner of the world at that time, and Svante was really humble in acknowledging the challenges to the ancient DNA, and suffering the number of defeats on his way he went through, and has been also largely responsible for the revolution of the high throughput methods in ancient DNA that have really changed the field and made it more democratic, more accessible to all of us. But I think we need also to acknowledge him for his contributions to the field of ancient hominins as a messenger of love, because when we think back in time when we didn't have DNA from the Neanderthals or the Denisovans, then we used to think about the differences between our species or the subspecies, if you like, in terms of more of conflict and war that humans colonized human Europe, and then the way they exterminated the Neanderthals. Whereas the messages from all of Svante's papers are messages of that whenever we met the other sorts of humans, we made love and we admixed, and then also when the Denisovans and the Neanderthals met, they made love and admixed. So that's a really a great message from his research. And then if you think about Tolkien, if he had been able to time travel, you can imagine how different the Lord of the Rings, the trilogy, would be without all these wars scenes. So that's really worth celebrating. But at the same time, the last year, 2002, also we need to remember as a very sad year because of the war in Ukraine, and all of us coming to this conference from Europe, probably having been in India before, recognize that when you sit on a plane, the plane journey takes longer because of the threat of Russia's war machines shooting down the planes. And we can't stay impartial about the war, not only because when Russia's troops are targeting schools and hospitals, but also because it's a war more generally addressed to all of us, because it is a war about the freedom of speech, about the freedom of press, and freedom of everything, basically, that is worth celebrating. And it is not only the freedom of speech being targeted in Russia, but also other dictators are banning freedom to speak, for example, about the evolution in Turkey since 2017. Teaching of evolution has been banned in Turkey, but nevertheless, since that year, the students of Ankara University hold every year meetings on evolution, where they invite delegates from across the world to teach them about evolution, regardless of the fact that the university or the state, support them. So this is something that we need to keep in mind, with that ethnicity and also race often are the terms that we have to confront when doing research on human diversity. And even though we may have thought that the distinction has been made already quite clear, then the same year, 2014, when Russia invaded Crimea, a book was published in America by Nicholas Wade, where the author claimed that genetic differences that are being observed among human populations have arisen recently due to selection for environmentally determined social behaviors, which is bringing us back a century to the thoughts of anthropologists of 19th century, and of course, or geneticists whose work the book cited responded and disagreed with Nicholas Wade. I think it is a moral duty for us working on evolutionary biology to keep on spreading the word and teaching the difference between ancestry and the reasons why the genetic there is no genetic base to support race. And we need to make a distinction between the terminology of relatedness, ancestry, and ethnicity and race to educate the students to avoid prejudice and discrimination. And it all starts from the principles of evolutionary biology, so with the need for us to define something that we use as a term. So when going back to Darwin, so there's been a lot of research on the definitions of what a species is, and also a lot of attempts are made to define diversity within human species. But if you start with phenotypic traits, then you can end up with either 4 or 20 classifications or more, and the underlying fact is that one phenotype by which you would define a group would not be correlated with the variation defined by another. So the phenotypic features that were used by anthropologists to define race were not leading to objective classifications that were aimed at to define race at individual level. And then also, when we think about the difference between race and ancestry, we need to think about the origins, and that is something that now with the tools from ancient DNA, we can go back in time and check that how diverse or how similar to the present day variation the groups were, and thanks to the advances of wide-scale Jiva studies, we can also look at the relationship between biology genes and behaviors and make a distinction also between those categories. But firstly, when we look at the human species, so then how much need is there to divide ourselves into meaningful, biologically meaningful groupings? It has been clear already for several decades that when we use the FSD as a measure of genetic differences, then humans do not stand out as a species with a very huge degree of differentiation. If you look at the other species like wolves or elephants even, then we see far greater within species diversity. Or when we look at the measure of heterozygosity, we see that compared to other great apes or even very narrow species with very narrow geographic range like pandas, we stand out as a species with very low level of differentiation. And then one interpretation of FSD given by Levontian is that if we see 15% on average in humans of FSD, then that means that most of the variation is embedded within population, and there's very little variation in between the populations. And the main explanation for these findings, even though Svante's work has been marvelously adding new nuances about the archaic admixture, I think the main picture of the out of Africa migration being the main factor explaining human genetic diversity and its gradual decline away from Africa is giving a demographic, recent demographic explanation to the human variation, namely that within less than 100,000 years ago, humans dispersed out of Africa, and therefore we see very little genetic differentiation among the groups. But also not only genetic, but also when we look at other traits like skull morphology, you see a similar decline of diversity away from Africa consistent with this model. And within regional areas such as West Eurasia or South Asia, we see relatively little differentiation compared to interregional variation, but even the interregional variation is somewhat very much minor to that what we observe in other species, and therefore we can say that the genetic differentiation in humans is fairly limited. And when we find genetic differences, we can often explain them also by geography, the geographic barriers. For example, if you go over Himalayas, then the genetic distances are greater than when you follow a region where there is no obvious geographic barrier, and also so we need to keep in mind that so if we want to define genetic differences at individual level, then when we look at the ancestry, then the differences are defined normally as mathematical probabilistic features, and typically when you look at individuals from a range of geographic regions, then you find each individual being composed of different ancestry components rather than being highly representative of a single ancestry. And these ancestry components, as we define them, are also to be considered multi-layered, so you can always continue finding substructure within them. So there is no natural way of defining at which level of structure to stop, which is quite in contrast to race and ethnicity, which are socially constructed terms, and often they are based on perceived characteristics. For example, Marlon Brando, who played an Italian character in a well-known movie. Himself is not Italian, but he's perceived as being Italian. And in a similar way, individuals can choose their ethnicities and also the race is somewhat subject to the definitions that are based on subjective terms. And that needs to be kept in mind when we use the word ancestry and the word ethnicity or race. And also, when we think about the terms of race, then quite important distinction also when we now look at the relationship between genotypes and phenotypes is that ancestry is a confounder to finding phenotype associations that we need to deal with, whereas when it comes to the definitions of race or ethnicity, then it is often perceived or assumed that your ancestry will determine what your phenotypes or behaviors are, and that's I think fundamentally the key distinction between those terms as how they should be used. But also when we talk about ancestry, often then we use also the words relatedness, and then for many of us, relatedness may refer to family trees and our relationship with individuals whose faces are familiar to us and then whom we know, whereas ancestry typically would relate to either country or continent-wide ancestry where we come from, and then but when you start thinking about the two terms, then there is no actual boundary between them, so it depends on how far back in time you want to go, and then at some point the ancestry and relatedness become also themes of the same underlying thing as where we come from. And then we need to note also that so often when we talk about ancestry, or sometimes also about relatedness, we refer to something very specific, and then and we refer to in singular, that one individual will have one ancestry, and then that ancestry is from one region, and that is to extend directly the issue with two loci. One is the Y chromosome and the other mitochondrial DNA, where indeed the variation is inherited in a surname-like fashion from one parent only to the offspring, and where we can see regional differences between West Asian, South Asian, African American populations in the frequencies of in this case the Y chromosome, or in case of this tree the mitochondrial DNA variation. But typically within regions, these systems provide very little resolution. So if you're from Europe, regardless of which parts of Europe you are almost likely to carry very similar haplogroup frequencies. So the thing, knowing your birthplace, you cannot predict what haplogroup you would carry. And the same would apply to India. India is super diverse, and then and with its own unique lineages, but knowing that a person comes from India would not enable us to predict that ancestry. But when we take a look at the autosomal genes, then we face a much more complex scenario, that we actually inherit genetic material from multiple parents, and that is actually one of the reasons, as it was mentioned earlier today, that by doing only one genome from ancient DNA can give you some glimpse of population ancestry, because each individual, each one of us, represents the diversity of many ancestors, rather than a single one of them. And that ancestry, if you go from the parent to offspring, can mean that distribution of signal. For example, in this case, the mother carries a red island signal, which is somewhat diluted in the genetic ancestry of the child. And even though I'm giving you here an example of diverse ancestries within a small region such as Estonia, you may think that this is quite ridiculous that all individuals have Estonian ancestry, but this is indeed a matter of scale. So the same principle applies if you think about much greater country like India, or if you take a bigger global view of ancestries as now with international travel. So the interbreeding between continents is quite common. But one of the assumptions, so that would be the future trajectory, that in the future we would expect to see more and more diversity in ancestry. But what about the assumption that when we go back in time, so with ancient DNA, do we see less diversity? And the answer is no. That Europe is particularly well covered by ancient DNA studies, and whichever time period you take, the outcoming messages that has been always a great deal of population movement, replacement, and admixture, and all European individuals, even though they may come up at global admixture plots as having a unique European ancestry. So when you go back in time, it shows that this is a composite of three different ancestries: Western hunter-gatherer, Eastern early farmer, and Yamnaya steppe ancestry. And within each group of them, you can also see sub-ancestry. For example, the Yamnaya ancestry is a composite of Eastern hunter-gatherer and Iranian hunter-gatherer ancestry. So when we go back in time, so we see more diversity, and also we can see that the underlying basis for a lot of variation that we see in modern populations today can be attributed to different groups. In case of Europe, so this is a reconstructed face of a Cheddar man from Britain, who was earlier, before ancient DNA studies, portrayed as a white man, similar to a local school teacher, but now from DNA studies we know that he had was probably a dark complexion, but blue eyes from the study by Brace et al. But also the study by Marnetto et al. has shown that this is not only the features of pigmentation or eye color, but a number of other traits can be also effectively assigned to co-vary with these ancient ancestries. And what is remarkable here is not only that there are features that can be positively assigned to those groups, but there are also a large number of traits that cannot be segregated between those ancestries. Again, supporting the view that the interrelationship between phenotype and genotype is far more complex than rather than due to packages of phenotypes deriving from a single ancestry group. And we know for many of those variants where we can actually establish the connection between the phenotype and the genotype, and that which are among the most differentiated SNPs, that the reason why they are highly differentiated between human populations is not the racial history, but it is the recent selection that has been driving these relationships. And we know, for example, that many those highly differentiated genes and SNPs are related to pigmentation, and then so with pigmentation, there is a relationship between the UV radiation, and then when we look at the whole genome data for the variants that have a deleterious effect on the genes, then even though we don't find a whole genome-wide signal of higher genetic load or higher rates of purified selection in non-African populations as predicted by some models, we see that in specific gene sets, the one like defined by 32 pigmentation genes, we find a significant excess of diversity on deleterious sites in non-African populations compared to Africans, which is consistent with a purifying selection for the UV damage protecting genes in tropical environments, and then also from the work conducted by Chandana and Mircea, we know that some of these genes that underlie pigmentation differences between Africans and Europeans are highly variable in India, and from Bullock's work we know that there is a difference between the different pigmentation genes, that the main contributor to variation in India as well as in between Europe and Africa, SLC24A5 gene apparently is allowed to vary as it maybe is not providing increased vulnerability to UV radiation, whereas other pigmentation genes like SLC45A2 has a restricted spread in India in relation to its the background genomic ancestry that provides us a prediction as how often you would expect to see certain genetic variants, and then in genes where there is a more the deleterious variation is expected to be more harmful in high UV conditions, you would see less diversity, and but in general, so when we look at the genetic ancestry, then apart from those variants that are associated with selection or pigmentation, in this case, what we could predict from the birthplace, basically a genotype. So if you're born in Europe, then we know that the 99% or more of Europeans have this particular SLC24A5 variant, and then we can predict the genotype. But this is not the case when we do a normal ancestry analysis. With given a large enough number of SNPs, we can quite effectively predict the birthplace from genetic data. But since we are dealing with common variants, we cannot do the reverse. So when we go back to any single common SNPs, then the frequencies between populations are fairly similar, and therefore we can't predict the genotype knowing the global ancestry group. And also, this is because most of the variation is shared between continents, and this is due to the large recent out of Africa migration that we share so much ancestry and common variants. And the variants that are private tend to be fairly restricted in their the distribution. But again, so when having a large enough number of rare variants, we can detect the clusters, but we can't do the opposite. We can't go back from the clusters to predict effectively at the individual level the genotypes. And there's of course also the cladistic problem of race, that we can't, when we put together the tree based on genetic sequences, we can't cut it in any meaningful way so that it would produce a single monogenic group of African population any point in the tree where we cut it, we will have some African populations segregating together with non-African populations, and that represents a problem for any meaningful biological analysis of the racial categories. And even though it has been shown for quite some time that there is a conflict between ancestry and race, yet in modern medicine in many countries still the self-reported racial categories are being used, and some proponents say that they provide some benefits for the diagnosis, but the overwhelming argument goes against it, because if it's not biologically well defined, then that means that we are more likely to provide misdiagnosis, and actually there's a growing evidence that in the following of those racially adjusted algorithms would lead to more discrimination and disservice to minority groups. But yet, so we need to consider the ancestry in population genetic studies, because if we don't do it, as I mentioned before, we end up having our results ancestry confounded, which basically means that we will assign some of the variation that is due to demography to the functional categories in a wrong way. And then just to visualize here a slide from someone Wilmer paper emphasizes the situation where you have collected controls and cases from a region, and then where the cases come from only a small sub-narrow parts of the region, and then the controls from the wider region would drive your difference between the cases and controls far apart, and then adjusting this by doing a locally matched case control study would significantly improve the situation, but also we are facing another downside of the exclusion of samples because of the non-matching status, particularly in Europe, by studies that typically remove all non-European ancestry individuals and carry out research only on those with European ancestry. So that means really that there's a crying need for more data from populations of non-European ancestry to remedy this situation. But this is not only the question about the global continental ancestries. As I mentioned at one of the earlier slides, there is no sharp boundary between relatedness and ancestry, and there is an increasing use of methods that take into account the haplotype of the IVD as a factor that is used in defining the structural units of population, and so at some point when we look at the more distant ancestors and individuals who share our distant ancestry, we can think of IVD also providing us a mean to query the structure of populations by simply considering the probability of IVD relationships between all pairs of individuals in a sample data set, and then by groups that can be either defined by geography or by unsupervised cluster analysis. So here is an example of a method that is highly scalable that was developed to study the connectivities within Belgian telephone users identify the modularities of connections by the number of calls made to other users and also the duration of the calls. And then what comes out of perhaps unsurprisingly is a very ethnically segregated picture of Dutch speakers speaking more to other Dutch speakers and French speakers to other French speakers. But that method can be also applied on genetic data if we consider the IBD as a signal of a connection, and you can apply the Leuven method to extract communities from genetic data and then combine large biobank data with medieval or pre-medieval genomes to map their ancestry. And we've done this on Estonian material, and then we get quite high resolution picture with a good correlation between the communities extracted by Leuven method and the geography. And what is also notable is that medieval individuals shown here by these boxes tend to map closely to the modern biobank individuals from the same regions. And that is true to the different medieval samples, whereas when we go back to Iron Age and Bronze Age, we lose the signal, and so we don't have any more connectivity, which sets a number of restrictions to the further analysis. And one question is whether this could be also the population structure that in different world regions may be translated from different time periods to time periods quite differently. So and that may also have consequences on biobanking information usage in different situations. So one of the projects that I've been recently involved in is related with the spread of plague in European populations, in particularly the second plague pandemic in the middle of the 14th century. And what is interesting is that in different European studies that have been performed in the last year or so, the outcome is quite different. So the Trondheim study here showed that the plague had quite a drastic effect on human mobility. That before a plague, individuals of quite wide ancestry in Europe, from British Isles and continental Europe, were found in the Trondheim samples, whereas when plague happened, then the city decided to pose a quarantine and close the city gates, and the long-term effect after that was that the city became more provincial, and then only individuals of local ancestry are represented in longer-term samples. On the other hand, another study conducted by Klonk et al. Found in a London cohort a huge difference between the variants that are associated with immunity. So before and after plague, a major selection event happened. Our study on Cambridge cohort could not reveal anything drastic in terms of the mobility of individuals associated with the 14th century event. Neither could we find evidence for the immune signal, but so consistent with many other studies, we find that HLA region has the highest heterozygosity. But in contrast to the Klonk et al. Study, we do not find the support for plague having a major immune effect on the population, even though when we use exactly the same set of neutral references that from the Bruno et al. Study that Klonk et al. Used, they use less than 10% of the neutral variants from that study. We we are able to replicate the results, but when using wider range of controls, neutral regions, we find no excess of FSD in Cambridge. And if you think about the effect, so our findings would rule out the polygenic effect on immunity of plague, whereas it can be still the case that plague affected a specific immune loci, but our screening of all immune variants would not be able to detect it. But also when we think about COVID or we think about the plague, if it's a very short-term effect, we probably do not expect to find a major effect on large number of genes as natural selection normally requires more time and more exposure from a disease to be infected. But finally, within this Cambridge cohort study, when applying the Leuven method on the People of British Isles project, the modern data, and then trying to map the medieval Cambridge samples onto it, we found that within modern British data, we can make quite good distinction between some regional populations, including from South Yorkshire, North Yorkshire, which the fine structure method in the Leslie et al. Paper, for example, couldn't clearly separate so well. But similarly to the previous study by Leslie et al., the main central part of England remains separable to us, and this is where most of the Cambridge samples fall, which probably tells us that the population movements during the history have quite significantly reshaped the genetic landscape. And then when we look at the correlations between those individual IBD-defined scores over time between different regions, we find that the medieval population is fairly highly correlated with present-day variation in England, but over time, so if we go back to the Roman or Saxon period samples from the same time, this is not the case. So the Roman and Saxon samples, similar to the Estonian material from Iron Age and Bronze Age, show very low level of correlation, and then suddenly within the medieval period, not only the English signal of correlation, but also the Dutch signal goes up, which is somewhat consistent with the historical records of a larger Dutch migrations to the east of England. And then if you were to use modern biobank data to study ancient remains from Britain, or from Cambridge in this case, if we had a Dutch biobank, it would serve as well as the English biobank for the purposes. And when we go from medieval to modern time, what we see is an increase of Welsh and Scottish ancestry, which is also consistent with population movements in the last few centuries, and these are probably mostly responsible for the mix-up of populations across Britain. But so when we do apply the UK Biobank data for the study of height as a polygenic trait in our sample, what is perhaps remarkable here is that recently a study by Prof showed that you can use the HSDNA to study the polygenic response, but the correlation between the height measured by femur or tibia, long bones in general, versus the predicted height from genes is only 6.3%. But that study involved ancient samples that cover a range of 850 to 33,000 years. So when we apply the PRS methods from UK Biobank data on medieval Cambridge, we see a 29% correlation, which is comparable to the estimates from modern data where the training and the validation samples come from different studies, highlighting the somewhat the limitations of this approach, that when we go back in time more than that our connectivity between the ancient and modern samples allows for, then we may lose the power of the modern biobanks, and then also that we probably would benefit from, in fact, of developing hand in hand not only the modern but also ancient biobanks with intensive phenotyping of the samples. So thank you very much. So there's a lot of people who have contributed to this.
Thank you very much, Toomas. Since this is a presidential lecture, we will not have any questions, but Toomas will be around, so if you have any questions, you'll be happy.
Thank you very much.
That's Urgen, and she is in Ladakhian dress, similar to the genetic diversity we explained here. We have very many students from different parts of the country studying in the institute.
Thank you.
Thank you, Toomas. Thank you, sir. So with this, we come to an end to the inaugural session and the presidential lecture. We'll now move towards the high tea, but before that, some announcements to be made. The speakers of the session 1 are kindly requested to give their presentation.